Again, good afternoon to all. So at this point, I will start discussing central dogma of molecular biology. And as we said, ang central dogma is comprised both of DNA replication and also gene expression process. When we talk of central dogma of molecular biology, this talks about the flow of information from the DNA going to RNA and finally in making a protein. So this figure summarizes the central dogma process in living organisms. As I've said, it all boils down, it all begins with the DNA, which will serve as a template, siya yung magsisilbing guide in the production of RNA, and that process is called transcription. Okay, that process is called transcription. And then ang RNA naman would serve as a template in the production finally of protein. So the term naman that we use to denote in making proteins, we have translation. Okay, translation. Okay? So yan ang central dogma. Particularly, magbibigay tayo ng emphasis sa tatlong processes. Number one, we have DNA replication, which will be discussed right now. And then for tomorrow, we have transcription and also translation. Collectively, as I've mentioned, ang transcription and translation are termed as gene. Anong tawag doon? Gene expression. Okay, gene expression. Tomorrow, magigets nyo bakit gene expression ang term. So ngayon, ang focus muna natin, we have DNA replication. So when we say DNA replication, as the term suggests, ang nakalagay, everyone, we have replication process. Basically, di ba pag sinabing replication, it means copying the original. Okay? Copying the original. Again, basically, pag sinabi nating replication per se, it simply means copying the original. So this means, again, this means that in replication, we tend to produce new DNA strand. Okay? We tend to produce new DNA strand such that, if you can still follow me, such that this new DNA strand is said to be complementary. Okay, this is said to be complementary to the parent. Okay, complementary to the parent DNA strand. As I've said, it is said to be complementary to the parent strand. And when we say parent strand, I repeat, when we say parent strand, we are also referring here to the okay, original strand, or what you call as the template strand. Okay, what you call as the template strand. So I reiterate, replication simply means copying the original, wherein we are producing new DNA strand that is complementary to the parent or the template strand. So for example, nasusundan ako, ha? Nasusundan? Okay, for example, if this is the template, makinig, if this is the template strand, having sequence, for instance, of A, T, C, and then G, okay, A, T, C, G, and if the arrangement is from 5' to 3', take note that this template strand would be copied, pero ang product would be complementary to it. So since meron kayo natutunan na complementarity rule, alam natin, i-expect natin ang produkto. So that would become T, nasusundan, T, and then we have A, and then G, and then we have C. And finally, the daughter strand or the product strand is anti-parallel based on Watson and Crick. Kaya it becomes 3' to 5' from top to bottom. Okay? So yan yung ibig sabihin natin ng DNA replication process. Yung introduction niya. Okay? So the original DNA molecule forms two new DNA molecules, each of which contains a strand from a parent and one new strand. So let's discuss rather deeper the process called DNA replication. One distinct property of DNA replication process is that it acts in a semi-conservative way. Again, it acts through a semi-conservative process or semi-conservative way. So this means that the old strand splits apart and acts as a template. So for you to understand further the semi-conservative process held by DNA replication, kindly refer everyone to the figure on the right side. Okay? Kindly refer on the figure sa right side. Suppose, nakikinig pa ba? Nakikinig? Okay? Suppose that the double helical structure found above is the parental double helix. Okay? Parental double helix. Take note that prerequisite for DNA replication is that this double-stranded parental strand, again, this double-stranded parental helix must separate from one another, okay? Must separate. And that process is called unwinding, okay? Unwinding process, also known as unzipping, okay? Unzip, okay? Hiwalay. That's why you can see red-colored structures, okay? Yung red-colored structures, these are the single-stranded parental strands. Okay, so these are the single-stranded parental DNA strands. Okay, so naghiwalay na sila. So ngayon, kapag naghiwalay na due to unwinding, this single-stranded parental DNA structure would serve as that template strand. Okay, template strand. Sila yung template. Sila yung pagkokopyahan para gumawa ng panibagong DNA strand or the products themselves. So in this case, ang nagiging product natin, yung color gray. Okay, yung color gray. So ito yung nagiging product. And this product, yung color gray na strand, this is said to be complementary to the parental strand. Okay, complementary to the parental strand. So taken all together, can we continue by the way? Continue? Okay. So taken all together, take note everyone, at the end of the first round, pagkatapos ng unang round ng DNA replication, meron tayong dalawang daughter strands na nagagawa. This is the first daughter strand and this is the second daughter strand. Now, anong connection nito sa semi-conservative? Pag sinabi nating semi-conservative, pag chinek mo ang daughter strands, hindi sila 100% new strands. Half is old, half is new. When I say half is old, ibig sabihin kalahating side ng daughter strand ay galing sa parental helix, parental. And then yung kalahati ay totally new. So ito yung kalahati, yung color orange or color red, galing pa rin siya sa parental strand, and then yung gray naman, ito yung new strand. Kaya yung product ng DNA, half is old, half is new. Ang tawag doon, semi-conservative. Okay? Kaya siya semi-conservative, may kalahating bago, may kalahating luma. Pag conservative lang ang term, lahat bago. Okay, lahat bago. Yun yung distinct sa DNA replication. So nanggagaling pa rin siya sa roots, sa mismong pinakaluma, pinagmulan. Nagets? Naintindihan? Okay, half is old, half is new at the end of the first round of replication. So refresh lang natin, in DNA replication, in semi-conservative process, the two parental strands separate. Anong tawag sa process of separation? Unwinding or unzipping. Okay? And each makes a copy of itself. Not literally copy, but complementary copy of itself. Okay? Complementary. So after one round of replication, there are two daughter strands, two daughter DNAs, wherein kapag tinrace natin ang bawat DNA structure, half of the helical structure is old, coming from the parental strand, and half is new. Okay? So yun yung ibig sabihin ng first round ng DNA replication, semi-conservative. Now, palalimin natin siya ng konti. So recall tayo. Halimbawa, tingin kayo sa PowerPoint everyone, okay? Tingin muna sa PowerPoint, wala mo na magsusulat. So for example, ito yung parental duplex, okay, parental strand. After the first round of replication, after the first replication cycle, meron tayong dalawang daughter strands. Ito yung una, ito yung pangalawa. Pero makikita natin, semi-conservative siya. Kasi may kalahati na galing pa rin sa parental, may galing sa, or may totally new naman. Okay, semi-conservative. Now, paano kapag nangyari ang second round or second cycle ng DNA replication? Imagine natin, makinig, imagine natin, yung first daughter DNA, at yung second daughter DNA, nag-unzip. Okay, nag-unzip sila. Pag nag-unzip, question, ilan ang template strands nyo ngayon? Ilan? 4. Very good. 4. So kung 4 ang template strands, ilan ang kailangan komplementan? 4 din. Kaya question, at the end of the second round, how many daughter DNAs ang mapoproduce? How many daughter DNAs? Okay, daughter DNAs. Ha? Hindi, yung buo na. 4 lang. 1, 2, 3, and then 4. Okay? So, 4 at the end of the second round. 4. Pero my question here is, sa 4 na yan, meron pa rin bang semi-conservative in terms of characteristic? Yes. Ilan ang nagdi-display ng semi-conservative model? 2. Dalawa semi-conservative, 2 completely conservative. Okay? Completely new. Pero ang bottom line, hindi pwedeng maputol yung pinanggalingan na parental strand. Okay? So yan ang ibig sabihin ng semi-conservative. Distinct yan sa DNA replication. Everyone, are you getting me so far? Okay, very good. So nasa tip pa lang tayo ng iceberg. Okay, palalalimin natin siya ng palalalimin as we go along. So yan ang DNA replication. Right now, the question is, ano yung mga specific steps involved para mangyari, ang production ng complementary DNA. Let us now proceed to the different steps involved in DNA replication process. In summary, we have 3 major steps. Number 1 is initiation. And number 1 is initiation, followed by elongation. Again, followed by elongation process. And lastly, we have termination. I repeat, we have 3 major steps involved. These are initiation, followed by elongation, and lastly, we have termination. In each major step, there are certain events na dapat nating malaman. So during initiation, please take note, number 1 would be identification of origin of replication, and often this is being termed as ORI. Okay? So ang symbol natin dito is ORI. Yan muna yung keyword na tatandaan nyo. Okay? So ORI. Next, second step under initiation, we have formation of the so-called replication forks, replication forks, and also primers. And also we have primers. So in short, sa step 2, take note we have replication forks and then primers. So ORI, replication forks, and then primers. Next, on the third step under initiation, we have unwinding. Ano another term sa unwinding? Unzipping. Okay, unzipping of double-stranded DNA. So we have unwinding. So all of this are under initiation. Okay? Mamaya magiging sensible siya lahat. Next, we also have elongation. Second major step is elongation. So ano yung mga specific events that occur during elongation process? Number 1, we have initiation of DNA synthesis and elongation. Okay? So una we have elongation. Or then initiation of elongation. Next, we have RNA primer excision. Okay, RNA primer excision. Whenever you encounter the term excision, it means removal. Okay, it means removal. So mamaya magkakaroon siya ng sense also. So removal, and then last, we have formation of replication bubbles with ligation. So ang keyword natin sa step 6 under elongation is ligation. Meaning ng ligation is kinoconnect. Okay? And then last step, as I've said a while ago, we have termination. Okay, we have termination. So yan everyone ang iisa-isahin natin. So start tayo guys sa initiation. O recap tayo sa initiation. Okay, initiation. So letter O means ORI. Nasusundan ako. Ha? O means ORI. R, we have replication fork o kaya primer. And lastly, we have unwinding or unzipping. So ang bibigyan natin ng emphasis yung mga steps na to. Starting off with ORI. Okay, ORI. So the replication, remember guys, bago ko makalimutan, ang dinidiscuss ko sa mga steps sa ngayon, prokaryotes. Okay, prokaryotes. Like the E. coli bacteria. Later ko i-discuss ang eukaryotes. Okay? So first stop, we have identification of ORI. Remember, When we talk about prokaryotes, they have circular DNA. So halimbawa, ito yung DNA. Halimbawa, double helical structure yan. Okay? Circular DNA structure ng prokaryotes. Napakarami ng DNA. Okay, napakarami niya or napakahaba niya as we tell. Ngayon, ang gap na iisipin nyo dito, sa haba ng DNA na yan, sa haba ng DNA na meron ang E. coli, for example, saan magsisimula ang replication? Ano ang starting point? Hindi siya madedetermine. Now, para malaman natin, para ma-pinpoint natin kung anong part ang magsisimula ang replication, we have to detect the so-called ORI or origin of replication. So sa buong DNA structure na meron ng prokaryotes, meron tayong segment dyan, meron tayong short segment na puro 80 lang ang makikita. Pag sinabi nating 80, sunod-sunod siya na mga nucleotide pairs na puro adenine-thymine ang pairing. Adenine-thymine, adenine-thymine, adenine-thymine. Okay? So meron siyang region, halimbawa, ito yon, meron siyang region na puro 80 segments lang ang makikita. Question, anong natutunan nyo kahapon about 80 base pairing? Kapag ang isang pairing ay puro 80, kumusta ang base stacking interaction ng mga ito? Less or more base stacking interaction? Parang ito na yung nag-brown out eh. O ano, kapag puro 80 segments, kumusta yung base stacking interaction ng DNA? Pag puro 80, less or more? Less, okay? Less. So merong less base stacking, okay? Less base stacking interaction sa puro 80 segment. Ulitin natin, kapag reach in 80 segments, there is less base stacking interaction. So kapag less ang base stacking interaction, ulitin natin, pag less base stacking interaction, less stable siya. Okay? Less stable. So when we say less stable, madali siyang ma-break. Madaling ma-break ang bonds between adenine-thymine kapag nag-stacking na sila o nagpatong-patong. Kaya ang tendency, mabubuka yon, mabubuka. Okay? Question, unang tanong pala, nasusundan ako? Yun yung una kong tanong. Nasusundan? Okay. Pangalawang tanong, kapag less stable, madali siyang i-break. Question, sino ang magbe-break? Ang nagbe-break, ang responsible sa breakage na yan ay isang klase ng protein. And that is what you call as the DNAA protein. Again, ang tawag doon ay DNAA protein. Ganyan talaga ang sulat niya. Okay? Maliliit yung NA, tas capital ulit yung A. So I repeat, the responsible protein that would cause melting of the 80 segments here. We have the DNAA protein. Continue. Can we continue? Okay. So dito tayo sa sample figure. For instance, tingin muna sa PowerPoint, everyone. Halimbawa, this is the prokaryotic DNA. So ang gap nga na sinasabi ko dito, sa dami ng DNA structure na makikita nyo dyan, ang malaking tanong sa atin, saan mag-i-start ang replication? Thank God, sa haba ng DNA na yan, may portion na puro 80 nga. Ang tawag doon, ORI, origin of replication. And since reach ito sa 80 segment, aatakihin siya ng DNAA protein, kaya ang tendency, lolobo siya. Okay? Lolobo. Eto, lolobo. Nakuha nyo? Lolobo siya. Okay? Kasi nawala ang base stacking interaction. Now, anong pinagkaiba ng ORI sa prokaryote at eukaryote? Tandaan, pag prokaryote, isa lang ang ORI. Okay? Kapag prokaryote, isa lang ang ORI. Kapag eukaryote, multiple ang ORIs. Ay multiple, marami ang ORIs. So prokaryote, isa lang ang ORI. And then kapag eukaryotes, multiple ang ORIs. So tayo, halimbawa, animal cells, marami tayong ORIs. Kaya as you can see in the structure or in the figure, sa left side, isa lang ang ORI kasi ito ay prokaryote, E. coli kasi bacteria ito, and then sa right side, multiple ang origin kasi nga eukaryotes ang nagre-represent dyan. So far, nasusundan ako? Ha? Pwede na tayong mag-continue? Okay, so since malalaman na natin kung saan ang starting point ng replication, what happens next? Sa step 2 ng initiation, ano ang keyword nyo? Ano yung R? Replication.
Replication fork.
Very good. It's replication fork. So please observe this structure ulit. Okay, this structure. So focus tayo sa prokaryotes. Kapag lumobo ang origin of replication, tendency may bubble na mangyayari. And take note, ang tawag natin sa boundary between this part, yung double-stranded structure, and yung isa na naghiwalay na single-stranded, ang tawag doon, replication fork. Okay? Replication fork ang tawag dyan. Huwag niyong pansinin yung color red ha. Huwag niyong pansinin yung color red na helical. Parang inassume dyan na meron ng kapares. Ang tignan nyo lang yung color blue. Okay? So sa pagitan ng double-stranded portion at ng single-stranded, ang tawag doon, replication fork. Okay? Replication fork. So anong nire-represent guys ng replication fork? Ang replication fork, this serves as, or they serve as, kasi dalawa. They serve as the actual site of DNA copying or DNA replication. So diyan mismo magaganap ang mga succeeding events para mag-replicate ang ating DNA or yung complementary strand para mabuo siya. Okay? So for example, ulitin lang natin, etong figure ulit na ito, imagine, pag lumobo siya at nagkaroon ng bubble, this part, okay, this part, ibahin natin kulay, okay, this part, sa pagitan ng double-stranded portions pa at ng single-stranded na hiniwalay because of the ORI, ang tawag doon, fork, okay, replication fork. And also this one represents a replication fork. Kaya in every ORI, may dalawang forks. Naintindihan ako? Okay, in every ORI, there are 2 forks. So dyan mismo nagaganap ang replication. Can we continue? Pwede na? Okay. Mamaya may papakita akong video sa inyo para mas ma-visualize nyo. Okay? So imagine muna sa ngayon, so imagine natin, kapag na-detect na ang replication forks dito, ang susunod na gagawin, palakihin ang butas. Palakihin yung bubble para magsilbi na template strand yung mga ibubuka. So para maibuka natin yon, kailangan maganap ang tinatawag na letter U. Anong tawag sa letter U? Kailangan ibuka.
Unwinding.
Very good. That is the process called unwinding, also known as unzipping process. Kaya ang step number 3, it involves unwinding or unzipping, okay? Unzipping process. So in unwinding or unzipping process, we need an enzyme here. Okay, we need an enzyme, and the enzyme responsible for the destruction or separation, rather not destruction, separation of double-stranded DNA into a single-stranded ones are the helicases. Okay, DNA helicases. So helicases are the ones responsible in the separation of the double-stranded DNA para sila maging single-stranded. So for example, in the figure, nasusundan pa ba ako? Ha? So for example, in the figure, yung color green, this represents helicase. Helicase. So helicase will separate the double-stranded DNA para paghiwalayin sila at maging single-stranded ones. And take note, tandaan ha, ang helicase ay nag-a-act bidirectional. Bidirectional. Ibig sabihin ng bidirectional, balik tayo dito, tingin sa PowerPoint. Wala mo na magsusulat, tingin sa PowerPoint. Pag sinabi nating bidirectional, may helicase na pupunta rito para ibuka yung mga double strand sa part na yan at may helicase na tutungo dito para ibuka sa kabila. Okay? Bidirectional yon. Para bumuka ng bumuka. Kaya sa bawat replication fork, may isang helicase. Then bubuka siya. Na-imagine nyo? Ha? Na-imagine nyo? Okay. So balik tayo dito. So naibuka na siya. So what happens next? O continue natin. Ang problema dito, ang possible problem is that pag binuka ng DNA helicase, yung double-stranded DNA, yung mga single-stranded, I repeat, yung mga single-stranded na nahiwalay na, may tendency bumalik sa dati. They can rejoin, in other words. Okay, they can rejoin. So para mapigilan ang rejoining, ng mga single-stranded DNA, kailangan merong mag-lock sa kanila. May pumigil. Huy, wag na kayong bumalik. Binuka na kayo ni Boss Helicase. Wag na kayong magbubuhol ulit. Okay? So thankfully, merong taga-kapit. May taga-kapit. Ang tawag natin sa taga-kapit, yung mga color orange dyan, okay? Yung parang tanikala dyan, ang tawag dyan ay single-stranded binding proteins. Okay? So ang tawag doon, single-stranded binding proteins. Ang single-stranded binding proteins, they are responsible in keeping the single-stranded DNA na ready for replication. Hindi sila hahayaang bumalik ulit sa double strand kasi mawawalan din ng sense yung ginagawa ng helicase. Okay? Kaya nakalagay nga dyan, during replication within the fork, helix destabilizing proteins bind to single-stranded DNA regions preventing rejoining. Okay? That prevent rejoining. Gets? Na-gets? Ha? Okay. Mamaya i-recall natin ang initiation. Okay? Next, in summary, sa initiation process, meron tayo mga important proteins na kailangan. Na-mention ba natin kanina ang DNA protein? Yes or no? Yes. Ano ang tatandaan nyo sa DNA protein related sa ORI? Okay, ORI. Anong sinisira ng DNA protein sa ORI? Kasi nga sa ORI, maraming complementary nucleotides na CG or AT. AT. Ang AT ba ay mahina? Yes, okay. Mahina siya, kaya masisira agad siya ng DNA. And then, anong purpose ng single-stranded binding protein? Siya yung magkakapit sa single-stranded DNA upang hindi mag-rejoin. Okay, yun yung importance. And then, of course, we have the helicases. Helicases are enzymes that would separate double-stranded DNA into single-stranded ones that would serve as templates later on. Okay, kapag nag-produce na tayo ng bagong mga DNA. Okay? Next, another figure that shows the functions of these proteins. So halimbawa, nabanggit na natin, we have the DNA. So pupunta nga ang mga DNA sa ORI, ay sa ORI. And then kapag na-form na ang replication bubble, ay kapag may replication bubble na, may kakapit sa mga na-separate, ang tawag sa kanila ay mga single-stranded binding proteins. And then yung color blue naman dito, this one, anong tawag dito sa color blue na ine-encircle natin? Anong tawag? Yes, helicase. Okay, yan yung helicase enzyme. Okay, very good. So yung helicase enzyme. So yan, natapos natin so far ang initiation. So initiation, number 1, ano yung palatandaan sa first step? ORI, very good. Number 2, R. Yung R yung importante doon. Mamaya na yung primer. Okay, replication fork. Yung primer later natin madidiscuss. Very good. And then last, helicase or unwinding. Okay,
Unwinding.
Okay. Anong susunod na step, major step? Letter E. Ano yung letter E?
Elongation.
Okay, nice. Elongation. Pero, bago tayo pumunta sa elongation, may plot twist. Okay, meron kayong dapat malaman about initiation. So para ma-connect natin siya sa discussion, let's have an analogy. Okay, let's have an analogy. Ang DNA ay isang double-stranded helical structure. So during replication process, particularly during initiation, via helicase, nagkakaroon ng separation ang double-stranded DNA into single-stranded. Ngayon, meron siyang implikasyon, meron siyang effect sa DNA pag ito ang nangyayari. Okay? Kapag ito ang nangyayari. So upang magets siya, imagine niyo ang isang lubid o rope. Hindi ba ang rope, kapag in-unwind mo yung nasa right side, chances are, pwedeng mag-super coil yung other side due to torsion, okay? Due to extreme forces. So kapag in-apply natin siya sa DNA, ang problem dito is kapag nag-act ang helicase enzyme here, yung mga distal portions ng DNA na double-stranded, pwede sila magbuhol, resulting to interference during DNA replication. What I mean to say is, kapag nag-act ang helicase dito, pwede ma-stop ang helicase pagpunta rito because of pagbubuhol. Okay? Pagbubuhol. Because of extreme torsion. Kaya, kailangan solusyonan ang supercoiling. Kailangan siyang solusyonan. So thank God, meron tayong enzyme in our body responsible for relieving the supercoil. At ang enzyme responsible for relieving the supercoil are the topoisomerases. Okay? So again, ang responsible enzymes dito, everyone, ay topoisomerases. So kumbaga, ang gagawin ng topoisomerase, aalisin yung pagbubuhol para hindi ma-stop ang DNA replication. Okay? Para hindi siya ma-stop. Pwede tayo mag-proceed? Ha? Okay. So another figure, bago ko i-discuss ang topoisomerase, ito na rin yung sinabi ko kanina. So imagine, yung cylinder for instance ay yung helicase. Okay? Yung cylinder ay yung helicase. So as helicase moves, okay, as it moves, chances are there could be supercoils that could, I repeat, that could interfere. Okay? Interfere further processes involved in replication. Kaya this has to be relieved. And ang responsible for the removal of this supercoiling are the enzymes called topoisomerases. Okay, these are topoisomerases. Topoisomerase enzymes solve the problem caused by tension generated by winding or unwinding of DNA. Now, take note that topoisomerases have two activities. Meron silang dalawang actions. Number one, meron silang nuclease action. Pag sinabing nuclease action, they can cut temporarily the supercoiled strands. Puputulin muna nila in the meantime. Okay? And then after that, kapag na-relieve na ang supercoiling, when everything gets back to normal, ilaligate nila yun ulit. Okay? Ire-reseal nila yun ulit. Kaya ang second activity held by topoisomerases, we have ligase activity or ligation action. So yan yung dalawang actions of topoisomerase. They cut initially, and when the tension is relieved, babalik na ulit sa original. Ididikit na nila ulit. Now, when we talk of DNA topoisomerases, we have two isoforms. Okay, meron tayong two isoforms. So you have topoisomerase 1 and then topoisomerase 2. Madali lang ito malaman kapag topoisomerase 1, ang nuclease action, yung cutting action ng topoisomerase 1, isa lang ang kaya niyang i-cut. Okay? Isang strand lang ang kaya niyang i-cut. Dun sa supercoil portion, isa lang. Pero kapag topoisomerase 2, both strands are being cut. Okay? Again, kapag topoisomerase 1, isa lang ang makakat. Kaya dito sa figure, as you can see, isa lang ang nakakat. Nakikita ba? And then, pag topoisomerase 2, both strands ang nakakat in the meantime. Okay, in the meantime. Yun nga, kapag na-relieve na yung problema, babalik na ulit sa resealing process that is malaligate na ulit. Hanggang ma-solve yung pagbubuhol. Okay? So yan ang DNA topoisomerases. Can you follow? Okay, recap tayo ng DNA replication sa initiation. So you answer what is being asked bago tayo mag-proceed. Number 1, ano ang reach sa ORI? Anong segments ang reach? 80. Less or more interaction or base stacking? Less. Very good. Anong klaseng protein ang nagme-melt ng 80 segments?
DNA protein.
Very good. DNA protein. That's right. Okay, next. How many forks are formed?
2.
2. Right. Very good. And then, anong tawag kapag dalawang direksyon? Bidirectional. Okay. Okay, bidirectional. What enzyme is responsible for the unwinding? Okay, helicase. Helicase. Okay, helicase. What keeps the single-stranded? Ang labo ng tanong ko, no? Pero ano yung hindi ko ma-English pero ano yung nagme-maintain ng single-stranded sa na-separate ng helicase? Yes, okay, very good. Single-stranded binding protein. That's right. What else? Sino ang nagre-relieve ng supercoiling? Topoisomerase. Dalawa ba yung activities ng topoisomerase? Yes. Anong enzymic action ang responsible for resealing?
Ligase.
Ligase, very good. Pag cutting,
Nuclease.
nuclease. Pag
Action.
both strands are being cut, anong tawag?
Topoisomerase
2.
2.
Pag isa lang, 1. Okay,
1.
very good. So, tapos na tayo sa initiation. So, ngayon, punta tayo sa elongation. So, makikita nyo, hindi muna ako nag-PowerPoint. Okay, magpe-pen and board muna ako. And then, tsaka tayo magdi-discuss sa PowerPoint. Okay, para step by step. Elongation. Pwede na tayo mag-start sa elongation, guys? Okay. Mas mahirap ito ha, mas mahirap, so makinig kayo. Unang tanong muna, ano ang sabi ni Watson at ni Crick sa DNA? Single o double-stranded? Double. How's the arrangement? Parallel or anti-parallel?
Antiparallel.
Okay. Anong tawag sa original strand? Template o complementary? Template. Anong tawag sa product? Complementary. Okay? Huwag niyong kakalimutan yan. Gamit na gamit natin siya today. So proceed tayo sa elongation. As the term suggests, in elongation process, dito natin makikita na yung complementary strand ay mapoproduce na, mapapahaba na siya. Okay? Dito na natin makikita ang formation ng complementary, ay formation ng complementary DNA strand. Okay, dito natin sila mawi-witness. At ang enzyme responsible for that, makinig, ang enzyme responsible for that is called the DNA polymerase. Okay, so the enzyme responsible is what you call as DNA polymerase. As the term suggests, in polymerase, it is responsible for polymerization. Pararamihin niya ang nucleotides upang makabuo ultimately ng DNA. Okay? So yan yung enzyme. Kaya lang, tandaan, ang DNA polymerase ay maarting enzyme. Okay, maarte siyang enzyme. At hindi siya basta-basta nag-act doon sa site ng replication. Meron siyang prerequisite para siya maging active. Okay, meron siyang prerequisite para siya maging active. And that is, kailangan bago sumugod ang DNA polymerase doon sa site of replication, makita muna niya ang tinatawag na RNA primer. Okay? Makita muna niya ang RNA primer. So ang prerequisite ni polymerase, kailangan ma-detect niya ang RNA primer. Question, ano ba ang RNA primer? Common sense, ang RNA primer ay binubuo ng RNA, ay RNA. Particularly, short strand of nucleotides lang makikita sa RNA, and that is approximately 10 nucleotides lang ang makikita natin. RNA nucleotides. So ang prerequisite natin para maging active ang DNA polymerase, we need RNA primer formation, which is characterized by having approximately 10 RNA nucleotides. Okay? Nasusundan? Okay, continue natin. Another question. Okay, another question. Sir, saan manggagaling ang RNA primer? Siyempre, isi-synthesize siya. Gagawin siya ng ating katawan. Ang gumagawa ng RNA primer ay isang enzyme. Okay? Ang gumagawa ng RNA primer ay isang enzyme. And that enzyme is called the primase enzyme. I repeat, the enzyme responsible for the formation of the RNA primer is the primase enzyme. Kaya at this point, ang i-discuss natin, mga properties ng primase. Again, i-discuss natin right now ang characteristics ng RNA primase. Number one, ang primase ay isang enzyme that reads the template strand. Okay, this is an enzyme that reads the template strand in 3' to 5' direction. Again, ang primase is an enzyme that reads the template strand from 3' to 5' direction. Question, when we say template, are we referring to the product or the original strand? Okay,
Original.
old or original. That's right. Okay, old or original. So yan ang activity ng primase. Okay, ng primase. So anong ibig sabihin nung reads the template? Anong kinalaman yan sa synthesis of RNA primer? So say for example, ito na yung medyo complicated ha. So for example, we have here the magnified view. Okay, magnified view ng replication fork. Isang side ito. Okay, isang side. So halimbawa, ito yung replication fork. Okay, replication fork, first and then second. Binagnify natin ang first replication fork. So ito siya. Na-imagine nyo? Na-imagine? Okay, so ito yung replication fork. And let's say, this has an anti-parallel arrangement. For instance, this is from 3' to 5'. The old strand, I am referring to the old strand. And then yung isa naman ay 5' to 3'. Ay 5' to 3'. Question, if anti-parallel ang arrangement, paano mag-a-act ang primase? Sabi natin, ang babasahin niyang strand ay mula sa ikatlo hanggang ikalimang prime. So tingin tayo sa unang strand sa taas. Ito yung unang strand, yung 3 to 5'. I have a question. Kapag 3 to 5 magbasa ang primase, paano ang galaw ng primase? Is it away the replication fork or towards the replication fork? Take two. Take two. Okay, take two. Ulit. Halimbawa, ito yung replication fork. Makinig ito yung replication fork. Question, paano magbasa ang primase? Ang binabasa niyang template mula 3 hanggang 5. Ay mula 3 hanggang 5. So kung 3 to 5 ang binabasa niyang direction, paano gagalaw ang primase? Paano gagalaw ang primase? Papuntang replication fork o palayo ng replication fork?
Palayo.
Ha? Away. Very
Palayo.
good. Away. Away. Away siya magbabasa. Kaya mag-i-start siya from here. Makinig mag-i-start ang primase here. Okay? Kasi nga 3 to 5. Gagalaw siya papunta sa kanan. Okay? Upper right. Papunta siya. Kaya ang mafo-form na product is this one. So ito ang primer. Okay? Ito yung primer. Okay, yan yung mabubuong primer sa baba. Kasi complementary yun eh. Ang tawag dito ay yung primer. Yung short strand ng RNA. Okay? So siya gagawa niyan away from the fork. Next, doon naman tayo sa baba. Okay, doon naman tayo sa baba. So sa baba, paano ang galaw ng primer or ng primase enzyme? Is it okay, papunta sa replication fork. Kaya manggagaling siya sa dulo kasi nga 3 to 5 direction. 3 to 5 direction. Kaya ang galaw niya is papunta sa fork. Kaya yung product niya which is the primer, ganito ang direction. Okay? Ito yung primer, yung short nucleotide sequence. Okay? Yan yung primer. Continue natin. Can we continue, guys? Okay, so very good. I-describe natin ang primer. Okay? I-describe natin rather ang primer. Question, what is the arrangement? What is the sequence of the primer? What is its direction? Is it 5 to 3 or 3 to 5 prime direction? Yung product mismo, yung primer. Is it 5 to 3 or 3 to 5? Ano ang rule? Ano ang rule natin? Kapag gumawa ka ng strand, it must be complementary and anti-parallel. Okay? Kaya ang primer, we expect na ang product ay 5' to 3'. Okay? So the product or the primer must be from 5' to 3'. Kasi the template being copied is the other way around. Okay? The template being copied is the other way around. Okay? So yan ang RNA primer. Tuloy natin. Hoy, okay pa kayo? Ha? Tuloy natin? Okay, very good. So recall tayo bago mag-continue. Ang responsible for elongation is DNA polymerase. Pero ang kwento natin dito, it cannot act muna kasi kailangan niya ng prerequisite, and that is the presence of the RNA primer. Ang RNA primer ay maikling segment lang ng RNA, 10 pirasong nucleotides lang ito, na ang gagawa ay ang primase enzyme. Paano gumagalaw ang primase? Gagalaw ang primase That is, ang titignan niyang kokopyahan or template strand, ang direksyon ng template ay from 3 to 5. Hahanapin niya saang strand yung 3 at saan yung 5. Ang starting point niya ay 3', kaya dun siya didikit, dun siya kakapit. And then yung complementary na gagawin niya ay anti-parallel. Kaya ang tendency sa unang strand sa taas, away from the fork siya. Kapag naman yung nasa baba, towards the fork. Okay, towards the fork. So yan ang importance ng RNA primer. So next, so malaking check na tayo sa RNA primer. Pag meron ng RNA primer, ready na ang polymerase enzyme. Okay? So dito tayo ngayon sa polymerase. O i-draw ulit natin upang magets. Okay? So halimbawa, we have the replication fork. Nandyan pa ba kayo mga bata? Okay. So halimbawa, ito ang replication fork. And then, for instance, ano ba yung sinulat natin kanina na arrangement? Una yung 3'. So halimbawa, 3', 5'. Yung kulay berde dito ay yung template strand na binuka ng helicase. Okay? And then, for instance, kukulayan ko ng red ang primer. So halimbawa, itong short segment na to, ang primer. Okay? And then, short segment, ang primer. So pag nakita ng polymerase, punta na tayo kay polymerase DNA polymerase. Kapag na-detect na ng DNA polymerase yung, as we said, yung starting point o yung RNA primer, doon na siya mag-act. Kaya yung polymerase, pupuntahan niya yung primer, pupunta siya rito, okay? Pupunta siya rito. And then yung isa naman, pupunta rito, okay? Pupunta sa kabila. Kasi dudugtungan niya lang yan. Ano naman ang kinakabit ng DNA polymerase? Ang ididikit na niya o ang ikakabit na niya ay mga DNA nucleotides. Okay? Siya na yung magpapahaba. Okay? Siya na yung magpapahaba ng complementary sequence. Now, ano yung characteristic ng DNA polymerase? Basically, ang characteristic niya, just like the primase enzyme, ano ang binabasa niyang template? Is it 5 to 3 or 3 to 5? Template ha, template.
3 to 5.
Okay, it reads the template from 3 to 5' direction. Kaya nga it only says na dudugtungan niya lang yung primer. Okay? Kasi nga nagbabasa rin ang primase same direction. Okay? It reads from 3' to 5'. Kaya mangyayari diyan via polymerase dudugtungan na niya ng DNA nucleotides yung primer. So yan yung gagawin ng DNA polymerase. Kaya ang product ng DNA polymerase, dalawang klaseng strands. Okay? Dalawang klaseng strands. Ano yung dalawang klaseng strands na magagawa ng DNA polymerase? Isang away from the fork, isang towards the fork. Okay? Isang away from the fork or replication fork, at yung isa naman papuntang replication fork. O, recap ulit, baka naliligaw na po tayo. Gets ba bakit may away tsaka towards the replication fork dito? Gets? Okay, very good. Question, alin ang papunta sa replication fork? Yung letter A o yung letter B? Or letter A o letter B? Okay, letter B. Okay, letter B. And then yung away, it's letter A. Ang tawag natin sa strand that is or that forms away from the fork, ang tawag dito lagging strand. Okay, ang tawag dito ay lagging strand. So I repeat, kapag away from the fork, yung nagagawa palayo, okay, palayo ng fork, that is called the lagging strand. Ang lagging strand ay tinatawag ding Okazaki fragments. Okay, so ang tawag din dito ay Okazaki fragment. And then, kapag naman towards, it means leading strand. Okay, leading strand. I repeat, kapag away, it's lagging or Okazaki fragment. Kapag naman towards, it's the leading strand. Now, tandaan din, kapag lagging strand, as the term suggests, it is a discontinuous strand. Okay, this is a discontinuous strand. And then, Kapag naman leading, continuous. Okay? Kapag naman leading, it's continuous. Diyan pa? Okay, let's continue. Question. Ay meron ulit question dito. Sir, ang tanong po namin, bakit ang tawag sa isa ay leading or discontinuous at bakit yung isa, bakit ang tawag sa isa ay lagging or discontinuous at bakit yung isa leading or continuous? It tells something about the process of this enzyme and other enzymes. O tingin kayo sa board, tingin kayo dito sa board. So halimbawa, this is the replication fork. Okay? Imagine niyo muna ha, although may video mamaya. Halimbawa yung color black, this is the helicase. Okay? This is the helicase. Ang helicase ba ang nagbubuka nung double-stranded? Ha? Yes, very good. O, wala muna magsusulat. Ang hirap i-discuss kapag virtual. Meron kasi ako mga body movements dito para mag-gets. Pero anyway, tingin kayo sa taas. Okay, taas. Ito. Ako, paano ba to? Ala, paano to? Nasira yung drawing ko, ha? Dali. Dali lang. Nasira. Aha. Nasaan na yun? Namamagnify pala siya. O, ito na lang. Sige. Ayan. Kita nyo na? Ha? Okay. So, una, bakit lagging strand? Pag lagging, putol-putol o tuloy-tuloy? Okay, putol-putol. So halimbawa, ito yung helicase. Okay, ito yung helicase. Imagine niyo ang helicase, binubuka niya yung ibang double-stranded. Okay, binubuka niya yung iba. Pero ang galaw ng polymerase sa strand na to, palayo ng fork. So ang tendency, habang binubuka ng helicase yung part na ito, naiiwan kasi ang polymerase palayo. Kaya nagpuputol-putol siya. Unlike no nasa baba, which is the leading strand. Ang leading strand kasi, kung saan ang galaw ng helicase, kung saan siya papunta, sumusunod lang yung polymerase. Kaya kung ibubuka ito ng helicase, susundan lang siya ng polymerase, mapaparisan, it is said to be continuous. Okay, it is said to be continuous. Kaya yung isa na papunta sa fork ay continuous, at yung isa na palayo sa fork, discontinuous. Okay? Nagpuputol-putol kasi kapag lagging strand. Kasi opposite ang direction ng polymerase at ng helicase. Okay? So yun yung reason behind it. Don't worry, meron tayong video for that. Mas may imagine nyo. But sa ngayon, tandaan nyo muna the leading and the lagging strand. Okay, o recap tayo bago tayo mag-proceed sa PowerPoint. Again, recap tayo. Number 1, kapag continuous, is it away or towards the fork?
Away.
Toward.
Okay, towards. Okay, very good. And then kapag away, is it leading or lagging?
Lagging.
Lagging, very good.
Lagging.
Pag same direction ng helicase, is it leading or lagging?
Leading.
Okay, leading. Kasi hindi nga siya nagpuputol-putol, hindi naiiwan yung polymerase. Okay? Meron tayong video for that later. So ito na rin lahat yung mga diniscuss ko so far. So number 1, initiation, we need the primer. Anong sabi natin sa primer? Short segment lang ba to na nucleotides? Yes. Sinong gumagawa? DNA polymerase o primase? Primase. How does it read the template strand? Is it 3 to 5 or 5 to 3? 3 to 5. How's the product? Is it 5 to 3 or 3 to 5?
5 to 3.
5 to 3. Anti-parallel. Okay, anti-parallel. Kapag may primer, dun nagagalaw ang polymerase. Question, is the movement of polymerase similar to the movement of primase? Yes, okay. Yes, tandaan nyo, it's a yes. Okay? So, tandaan, kapag same direction, edi may starting point na tayo. Now, sa primer, ito na yung sinabi ko, basahin nyo na lang to, okay? Ito na yung diniscuss ko lahat. Cannot initiate synthesis, kailangan natin ng a short piece of RNA. Okay? That is the RNA primer. So ang RNA primer ay isang short segment of nucleotides na ginagawa ng primase. O tignan nyo yung picture. Makinig, tignan nyo yung picture. O di ba? Na-draw natin siya. Kaya lang dito kasi ang nasa taas 5 to 3 and nasa baba 3 to 5. Basta alam nyo kung paano magbasa ang primase, you will understand this. Okay? Hindi kayo mawawala, hindi kayo maliligaw. Kaya you can see na yung primer dito sa figure, again, yung primer sa figure, ito siya, yung color green. Yung color green, yan yung primer. Kasi nga, from 3 to 5 ang basa ng template. Okay? And then we have here, ito naman. Now, kapag nag-act na ang polymerase enzyme, di ba sabi natin ang polymerase ay dudugtungan niya lang yung primer. So for instance, this is the polymerase. Halimbawa, ito yung polymerase, yung color green na dinodrawing natin, and this one is the polymerase. And then, yung color pink, for example, yung helicase. Ito yung sinasabi ko kanina na kapag in-unwind or in-unzip ng helicase yung mga part na to, at pag bumuka siya, ang tendency, yung nasa taas, that is towards the fork, it's continuous. Kasi sinusundan niya lang yung helicase. Nagigets niyo ba ako? Okay? Sinusundan niya lang yung fork. Kaya it's continuous. And then yung nasa baba, it's lagging. Kasi pag gumalaw ang polymerase pababa, pag gumalaw siya, at bumuka na naman ito, wala siyang primer. Gagawa na naman ng panibago. Kaya primer, then polymerase, kaya putol-putol siya. Anong tawag sa putol-putol? Lagging strand, also known as Okazaki fragment. Okay, Okazaki fragment. So yan ay mga pinagsasabi ko kanina. O ito rin, diniscuss ko na to. Responsible, polymerase na po tayo, Paano siya magbasa ng template? Tama bang 3 to 5 ang basa niya ng template?
Yes.
Yes, very good. At ano ang product niya? 5 to 3. Very good. So yan yung ating diniscuss kanina. And then, this one, iba lang ng presentation pero yun pa din siya. Ang general rule, anti-parallel. So we have the leading strand, o recall tayo pag leading. Same direction ba ng helicase? Yes, papunta sa fork. Papunta sa fork. Okay. Kapag lagging, palayo, opposite direction. Question, sa lagging strand ba, do we need multiple primers here? Yes or no? Kailangan ba natin na maraming primers sa lagging? Yes. Okay? Kasi nga naiiwan. Kaya kailangan gumalaw na naman. Okay? So yan ang important. So ito na lahat ng mga sinabi ko. O di ba? Nadoble lang. Nadoble lang. Pero kasi mas maganda kapag one step at a time. So at least ito, reinforcement na lang sa inyo. Pag advancing, palapit. Pag palapit, leading. Sir, paulit-ulit ka. Okay lang yon. And next, away, it's lagging strand, Okazaki fragment. Okay? Okazaki fragment. Can you follow? Okay. So, nangangalahati na tayo sa polymerase. Meron pang ibang actions ang polymerase. So mag-focus tayo everyone pa rin sa DNA polymerase. Let me tell you, class, na meron tayong dalawang important properties held by polymerases. Number one, they have the ability to elongate as we discussed a while ago. And one distinct property of DNA polymerase, they can double check what they did. Okay? Pwede nilang i-double check yung mga ginawa nila kung tama ba yon or mali. So ang tawag doon ay proofreading. So when we connect this with their enzymic actions, tandaan, ang elongation or chain elongation is it's because of the polymerase action of the DNA polymerases. So ulitin natin, ang chain elongation and processivity, this is due to the polymerase action by DNA polymerase. Whereas, ang proofreading naman, again, ang proofreading naman is caused by the exonuclease action held by DNA polymerases. So ulitin natin, meron silang polymerase action, elongation yon, pag double check, exonuclease. Okay? Exonuclease. So unahin natin, okay? Unahin natin i-discuss ang iba't ibang types ng polymerase. So meron tayong iba't ibang types ng polymerase enzyme. Dito tayo sa isang platform para mas malaki. For a while lang. Okay. So dito tayo sa different types of DNA polymerase. Okay, so everyone, let me tell you na we have two major types of DNA polymerases in prokaryotes. Okay, in prokaryotes. So number one, we have the DNA polymerase 3. Okay, so number one, we have the DNA polymerase 3. So meron tayong dalawang properties by DNA polymerase 3. So number one, it is responsible for the elongation of the DNA strand. Okay? So it is responsible for the elongation of the DNA strand. Ito rin yung diniscuss natin kanina when we discussed the leading and the lagging strand. And as we recall, ang DNA polymerase 3, okay, for example, is responsible for reading the template. It can read the template from 3 to 5 prime direction. That's why ang kanyang elongation action, that is from 5 prime to 3 prime. Kasi we are talking here about the product na kaya niyang gawin. Okay, we are talking here about the product na kaya niyang gawin. So that is yung complementary strand. So that's why, because of its elongation activity, it has a 5 prime to 3 prime polymerase action. Okay? It has 5 prime to 3 prime polymerase action. So yan yung unang activity ng polymerase kasi it can elongate the DNA strand from 5 to 3. So tawag doon DNA or 5 to 3 polymerase action. So for example, i-draw natin. Okay, for example, this is the original strand, okay, o yung template strand or the original strand. And then meron tayong sequence like A, and then T, and then C, and then G. So the direction, as I've said, for the original strand is from 3 prime to 5 prime. Okay, so ang direction is from 3 prime to 5 prime. Kaya ang polymerase, ang DNA polymerase 3, ang gagawin niyang strand, that is T muna. Okay, T, halimbawa ito yon, yung ginawa niya rather, that is T, and then A, and then G, and then C. Okay? So ang naging direction ng polymerase 3 is from left to right. Okay, left to right. Kasi nga, it reads the template 3 to 5. And then the product is from 5 to 3. Okay, the product is from 5 to 3. Kaya meron siyang 5 to 3 elongation action or polymerase action. Okay, so yan yung una niyang activity, that is elongation. Next, meron pang isang kayang gawin ang polymerase 3, okay, polymerase 3. Pangalawa, kaya niyang gawin ang proofreading. Okay? Kaya niyang gawin ang proofreading activity. Pag sinabi natin proofreading or double check, ganito ang mangyayari. Kapag naglagay siya ng isang nucleotide, okay? Pag naglagay siya ng isang nucleotide, naka-complement nung parent strand, i-double check niya yon. Okay? Halimbawa, ako yung polymerase 3. Halimbawa, ako yung polymerase 3. Bawa, gumalaw ako. Pinarisan ko siya ng T. Paglipat ko sa kabila, bago ko maglagay ng panibago, lilingon ako sa kabila. Babalik ako, kumusta ka? Tama ba siya? So halimbawa, A yung nasa parent. Ako yung polymerase, and then nilagyan ko ng T halimbawa, yung A, nilagyan ko ng T. Paglipat ko sa kabila, babalikan ko yon at double check ko, tama ba yung nalagay ko o mali? Kasi kapag mali siya, papalitan niya yon ng tamang pair. Okay? Double checking ang tawag doon. So ang proofreading niya, tingin kayo dito sa figure natin, halimbawa yung polymerase nandito na. Ay halimbawa nandito na yung polymerase. Halimbawa polymerase 3 ito. Pagpunta niya sa side na to, after lagyan ng T, babalikan niya yon at check niya kung T ba talaga ang nalagay. In the event na ang nalagay niya ay C, at bumalik siya, na-realize niya mali, aalisin niya yon, lalagyan niya ng T talaga. Dino-double check. Kaya ang proofreading activity niya, pabalik nung template, pabalik nung complementary na ginawa niya. Kaya meron siyang 3 prime to 5 prime Kasi pabalik eh. Nakukuha nyo, 3 to 5. 3 to 5 prime. So pag proofread, anong enzymic activity? Polymerase o exonuclease? Okay, so meron siyang 3 prime to 5 prime exonuclease action. Okay? So meron siya ngayong 3 prime to 5 prime exonuclease action. Gets? Ha? Okay. So recap tayo sa PowerPoint. So ulitin natin yung unang type ng DNA polymerase na discuss ko. It's DNA polymerase 3. Dalawa ang actions ng DNA polymerase 3. It has elongation and it has proofreading. So sa elongation, as I've said, meron siyang 5' to 3' polymerase action. Okay? Meron siyang 5' to 3' polymerase action. Okay? Kasi nga ang daughter strand na nagagawa niya ay anti-parallel. Okay? Anti-parallel. So ito na yung sinabi ko kanina na illustration natin about the action of DNA polymerase 3. And then, one distinct property also of DNA polymerase 3, it can double check kung ano yung natapos niya, kung ano yung nagawa niya. So sabi dito, double checking of nucleotide after it is added. So yun nga, pag pwesto ng isang nucleotide, paglipat sa kabila, babalikan na naman. Tama ba to o mali? So since babalikan, dapat ang movement ng polymerase kabaliktaran nung kanina. Kaya it becomes 3' to 5' exonuclease action. Okay? 3' to 5' exonuclease action. So for example, o tingin tayo sa PowerPoint, ay for example in this figure, so suppose we have here, yung color blue is the original strand, okay, original strand, and then yung color orange is yung complementary strand. So halimbawa, nakapwesto na dito ang polymerase 3. So nagpares siya. Ang pinares daw niya sa T, nakikinig pa ba kayo? Ha? Ang pinares niya raw sa thymine ay cytosine. Tama ba 'yon? Mali. Okay, so tingnan natin. Pagpunta niya dito, iniwan niya yung C. Paglipat niya sa kabila, binalikan niya. Okay, binalikan niya. Kaya ang activity niya is pabalik, it becomes 3 to 5' kasi 3 pabalik kasi binalikan niya eh. Okay? Ang relativity na pinag-uusapan ko, whether polymerase or exonuclease, sa product. Okay, sa product kayo titingin. Kaya binalikan niya, inalis niya, kaya exonuclease, okay, exonuclease action. And instead, it was replaced by the correct base pair, and it must be adenine. Kaya makikita nyo sa dulo, it already becomes adenine. Okay, it became adenine. And then pag napera na ng tama, okay na, pwede na niyang iwan yun ulit. Okay, kaya nga strictly regulated ang DNA replication kasi otherwise that could lead to disease, mutation sa madaling salita. Okay? So yan ang proofreading activity. So natapos natin ang DNA polymerase 3. Okay, natapos natin ang DNA polymerase 3. Question ulit, sa DNA polymerase 3, meron ba siyang elongation activity? Yes. Ano ang product ng elongation niya? 5 to 3 or 3 to 5? 5 to 3, very good. Nagdo-double check ba si 3?
Yes.
Paano siya
Yes.
paano siya nagdo-double check? Opposite direction ba nung complementary? Yes. Kaya it's 3 to 5. Pag kapag double check, is it polymerase or exonuclease? Okay, exonuclease. Kaya ang polymerase 3, meron siyang 5' to 3' polymerase at meron siyang 3' to 5' exonuclease action. Okay? Exonuclease. Hindi ko pala to naalis, no? Dapat naalisin ko to eh. Kasi last year, nung tinuro ko to, sunod-sunod talaga yung mga diniscuss namin. Kaya nalagay ko to. Ewan ko ba kung anong sapa ko sa ulot, nasama ko yan. Anyway, feeling ko hindi pa naman, sabaw na ba kayo? Ha? Hindi pa. Very good. Okay. So, after natin sa DNA polymerase 3, may isa pang klase ng polymerase. Okay? Meron pang isang klase ng polymerase. That is DNA polymerase 1. Okay? So, ano ang function ng DNA polymerase 1? Ang DNA polymerase 1 is responsible for the excision of RNA primer. Question, importante ba ang RNA primer para maging active ang polymerase in general? Yes. Pero hindi habang buhay, dapat nandun ang primer. There will come a time that this RNA must be removed. Okay? It must be removed. Kaya nga excision means removal of the primer. Okay? Aalisin siya. At ang nag-aalis doon ay si DNA polymerase 1. Okay? At nag-aalis doon ay si DNA polymerase 1. Question, kapag nag-aalis o kapag nagre-remove, anong function yon? Polymerase o exonuclease? Okay, very good. Exonuclease. Therefore, ang polymerase 1, okay, therefore, ang polymerase 1, may exonuclease action din siya. Okay, may exonuclease. Pero, ano ang kanyang exonuclease action? Paano niya inaalis ang RNA primer? O recap tayo. Ano ang direksyon ng RNA primer? Ano direksyon ng RNA primer? 5 to 3 or 3 to 5? Anong RNA primer? Ito ba yung product? Product ba siya? Product siya, di ba? Yung short segment. So ano yung product ng RNA primer? 5 to 3 or 3 to 5 direction?
5 to 3.
Okay, very good. 5 to 3. Okay, 5 to 3 ang kanyang arrangement. Okay, 5 to 3. So for example, makinig ha. For example, ito yung short segment ng RNA. Halimbawa, U, A, U, C, U. Halimbawa, and so on and so forth. Yan yung short segment. From 5' to 3'. Yan yung primer. For example, a primer. So ano pong gagawin ng DNA polymerase 1? Ang DNA polymerase 1, siya yung magre-remove ng bawat nucleotide. Okay, magre-remove ng bawat nucleotide. At ang direksyon ng pag-alis niya mula 5 hanggang 3. So papunta dito, 5 to 3. Kaya meron siyang 5' to 3' exonuclease action. Okay? Kaya meron siyang 5' to 3' exonuclease action. Kasi it removes. Okay? Next, pagkatapos niyang alisin ang mga RNA nucleotides, anong gagawin niya? Papalitan niya yon ng DNA. Papalitan niya ng DNA nucleotides. At ang direksyon ng pagpalit ay 5 to 3 din. Okay, 5 to 3. Question, anong enzymic action pag nire-replace ng DNA? Yung pagpapahaba, is it exonuclease o polymerase?
Polymerase.
Okay, kaya meron siyang 5' to 3' polymerase action. Okay? Again, meron siyang 5' to 3' polymerase action. And finally, yung ni-replace niya, yung ipinalit niyang DNA nucleotide, nililingon niya ulit pabalik. Tama ba itong nailagay ko? Anong tawag kapag nililingon? Exonuclease. So kung nililingon, kabaliktaran ng direksyon yon. Kabaliktaran. So it becomes 3' to 5' exonuclease action. Okay? It becomes 3' to 5' exonuclease action. So that is the proofreading activity by polymerase 1. Okay, o review tayo ulit. Okay, review tayo ulit ng polymerase 1. Polymerase 1. Una, kailangan ba maalis ng primer? Yes. Anong arrangement ng primer? Ano ang direction ng primer? 5 to 3 or 3 to 5? 5 to 3.
5 to 3.
So kung ano man ang direction ng primer, ganun din ang sequence ng paggalaw ng polymerase 1 para alisin ang RNA. Kaya meron siyang pag-alis in 5 to 3 direction. Anong tawag pag-alis? Is it exonuclease or polymerase? Okay, exonuclease. Kaya meron siyang 5' to 3' exonuclease. And then, syempre may gap doon kasi tinanggal eh. Ay, kasi tinanggal. So pag tinanggal, papalitan ng DNA. Paano papalitan? 5 to 3 din. Kaya 5' to 3' polymerase action. Okay? And then, nagdo-double check ba si polymerase 1? Yes. So anong direction pag double checking?
3 to 5.
Okay, 3' to 5' exonuclease action. So another figure. Nandyan pa kayo? Ha? Okay. So next, another figure that would further explain what I said. So imagine, yung color red, makinig ha, makinig. So imagine, yung color red ay yung RNA primer. So ito yung RNA primer, yung color red dyan. And as we said, anong arrangement ng RNA primer? From 5 to 3. Okay? Aalisin siya ng polymerase 1 kaya natanggal. O, nakita nyo natanggal. Paano gumalaw ang polymerase 1 in removing the primer? That is, ang ginagawa niya, 5 to 3 exonuclease. Okay, 5 to 3. Kaya natanggal. And then question, ire-replace ba to ng panibagong DNA? Yes. Paano ang replacement? 3 to 5 or 5 to 3?
5 to 3.
5 to 3. Okay. Do-double check ba?
Yes.
Paano din-double check? 5 to 3, 3 to 5.
3 to 5 po.
Anong tawag? Polymerase, exonuclease?
Exonuclease.
Okay, very good. Okay. So, yan yung dalawang types ng DNA polymerase. So, okay pa ba kayo? Ha? Totoo, okay pa kayo? Kasi ako hindi na. Anyway, kidding aside, i-summarize natin. Okay, i-summarize natin. So in this table summarizes the enzymic actions of DNA polymerase 1 and 3. I-summarize natin, number 1, for DNA polymerase 1. DNA polymerase 1, alalahanin nyo, meron ba siyang 5' to 3' polymerase? Yes or no? Yes. Okay, yes. Anong specific process ito? Anong specific process ito? Yung 5 to 3 polymerase action. That is, yes, elongation. Elongation pero yung pag-replace nung natanggal. Ano ba yung natanggal kanina? Ano yung tinanggal nung 1 kanina? Primer. Very good yung primer. Okay? Next. Meron ba siyang 3' to 5' exonuclease? Yes. So ano yung 3' to 5' exonuclease? Is it the removal of primer or is it proofreading? Yes, it's proofreading. Okay? Next, 5' to 3' exonuclease. Is it for the removal of primer?
Removal.
Yes, okay. So check na tayo sa polymerase 1. Okay, check na tayo sa polymerase 1. Next, polymerase 3. Polymerase 3. Question, meron ba siyang 5' to 3' polymerase action? Yes. So saang portion siya merong ganitong function, that is elongation, yung pagpapahaba talaga ng DNA. Siya may responsible dun sa lahat, okay? Pagpapahaba. Next, meron ba siyang 3 to 5 exonuclease? Yes. Ito ba yung proofreading, mga bata? Okay. Question, can it remove RNA primer? No, it cannot remove. Kaya wala siyang 5 to 3 exonuclease action. Okay? Kaya wala siyang 5' to 3' exonuclease action. Okay? So overall, yan po ang function ng DNA polymerase. Okay? So sa DNA polymerase, diniscuss natin ang elongation process and also excision ng primer. Next, under pa rin tayo ng elongation, mga bata. Ang last step ng elongation is yung ligation. Okay? Ligation. Naaalala nyo ba yung Okazaki fragments kanina? Naalala nyo Okazaki fragments? Very good. Anong Okazaki? Putol-putol ba sila?
Yes.
Okay.
Yes.
Question, paano mapagdudugtong-dugtong ang mga Okazaki fragments? Kailangan natin ng ligase enzyme. Okay? It needs the ligase enzyme. So via DNA ligase, mako-connect ang mga lagging strands. O yun lang yung tandaan nyo. Okay? Yun lang yung function ng DNA ligase. So overall, tapos na po ang elongation. O review tayo ng elongation. Nandyan pa po ba?
Yes po.
Okay.
Opo.
So recall tayo ng elongation. So ang unang step sa elongation is initiation ng DNA synthesis. Anong napag-usapan natin dito? Kailangan ng primer. Kailangan ng primer na gagawin ng primase. At saka dudugtungan ng polymerase enzyme. Okay? Polymerase. Ngayon, bago matapos ang DNA replication, kailangan alisin ultimately ang RNA primer. Ayan o, kasusulat ko lang kanina, removal. Sino nag-aalis nito? Polymerase enzyme din ba ang nag-aalis ng primer? Yes. 1 o 3? 1. Okay? 1. Okay? So, it is DNA, DNA polymerase 1. And then, ligation. So tapos na. Ay, tapos na ang elongation. Parang ganun kadali, no? Pero tapos na ang elongation. And then finally, we have termination. Nasaan na tayo? Termination. So in termination, this simply means it marks the end of DNA replication process. So this is mediated by sequence-specific binding of terminus utilization substance to the replication sites, thus stopping the movement of the replication fork. So pag nag-stop ang replication fork or pag nag-stop ang action ng helicase, so hindi na magko-continue ang polymerization. Okay? Kaya wala na tayong DNA replication process. So yan ang termination. So altogether, natapos natin guys ang steps in DNA replication. O review tayo ulit mula umpisa. Okay, review tayo ulit mula umpisa. So dito sa figure na pinapakita, ito yung summary ng DNA replication process. So una, tingin kayo sa PowerPoint, yung color orange is the helicase. Okay, helicase. Ang helicase ba ang naghihiwalay? Yes. Okay? Anong tawag sa mga yellow? Anong tawag sa mga yellow na yan? Anong tawag dyan sa yellow? Sila ba'y nagme-maintain ng single-stranded? Yes. Okay. So single-stranded binding proteins. And then, actually hindi kompleto yun nasa figure, pero medyo kita naman ata siya. Okay? Halimbawa, etong nasa baba, yung putol-putol, this is the lagging strand. Okay? Okazaki fragments. Pag Okazaki, anong natutunan nyo? Palapit o palayo ng fork? Palayo. Very good. Putol-putol siya. Okay? And then, may primase tayo. Importante ang primase kasi prerequisite siya para ma-activate ang polymerase. Okay? Para ma-activate. And then, meron tayong ligase na nagdudugtong sa mga Okazaki fragments. Yung nasa taas, wala masyadong problema kasi sinusundan lang niya ang galaw ng replication fork. Okay? Helicase sa replication fork. Next, another review sa mga proteins involved in replication. Anong function ng DNA polymerase? Although na-discuss natin elaborately ito, so polymerization in general. Pero syempre, meron pa rin proofreading activity. Okay? May exonuclease activity pa rin. Helicase, unwinding. Ang topoisomerase ba, naalala nyo pa kanina? Ha? Anong function ng topoisomerase? To prevent
yung supercoiling.
Okay, very good. To treat or to prevent supercoiling. Ang primase ba ay important mga bata? Yes, para makabuo ng anong binubuo ng primase? Primer. Okay, RNA primer na gagamitin ng or napagbabasihan ng polymerase. Okay, napagbabasihan ng polymerase. Single-stranded binding proteins, okay na yan. And then, DNA ligase. So yan po ang mga proteins needed sa DNA replication. Are we learning so far? Are we still learning? Okay. So natapos natin ang DNA replication sa PowerPoint. So para makatch natin lahat ng mga students, if ever hindi ma-imagine nung iba yung mga sinasabi ko, meron tayong prepared video na pwede natin magamit para mas maintindihan. Okay? Wait lang guys, i-prepare lang natin yung video. Hindi na ako mag-break ha, para early dismissal tayo. Okay? Is it okay? Yes or yes? Okay lang? Sige. Para hindi tayo umabot ng 3:30. Anyway. Nakikita po? So ipopost ko to from time to time, tas i-explain ko. Okay? Para lang ma-reinforce yung mga sinabi natin. Sorry.
DNA is a molecule made up of two
Wait lang, naririnig? Naririnig? Sige.
strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands.
Actually, hindi lang na-mention dito sa video, pero may ORI dapat ha, may ORI. Anong tawag sa parating no enzyme? Parating na enzyme. Anong tawag dyan? Helicase. Very good.
This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA.
Huwag niyong kalimutan na yung helicase nasa side na to, okay? Nasa side na to. Kasi basis natin yan in understanding the leading and the lagging strands later.
An enzyme called primase starts the process.
Ang mag-i-start, hindi ba? Primase. Gumagawa ang primase ng RNA primer. Paano nagbabasa ang primase? Paano niya binabasa yung template? 3 to 5 o 5 to 3? Yung template, very good, 3 to 5. So saan siya pupunta? Papuntang fork o palayo ng fork? Pag nasa taas, okay? That is, papunta, okay.
This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end.
O para malinaw, yung sinabi sa video na it adds DNA bases from 5 to 3, it means the product. Okay, the product. Kaya nga, anong sabi ko kanina? Pag DNA polymerase sa elongation, ano ang kanyang polymerase action? It's 5 to 3 polymerase action. Nagigets? Ha? Okay, next.
One of the new strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand.
Gets nyo bakit putol-putol? Nagets nyo ba't putol-putol siya? Yes. Kasi nga, continuous ang galaw nung naghihiwalay. Sino yung naghihiwalay na enzyme? Si helicase. Okay, si helicase. So, galaw ng galaw ang helicase, may nagsisingle-stranded na naman, ang polymerase, opposite ng direction, may naiiwan. Kaya kailangan magkabit ulit, di ba kailangan ng primer? Kaya putol-putol siya. Lagging strand ang tawag doon.
Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands.
May sinabi dito sa video na exonuclease removes the primer. Actually, exonuclease ang sinabi pero polymerase enzyme pa rin yan. O anong polymerase yung nag-alis ng primer? Polymerase 1. Very good. 1. Paano siya mag-alis? 5 to 3 or 3 to 5? Yes, 5 to 3. Siya rin ba yung magre-replace nung DNA? Yes. Same direction ba? 5 to 3 din? I-double check niya ba? Paano siya mag-double check? Lingon. So pag lingon, 3 to 5. Very good.
Strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA.
O yan din yung polymerase 1.
Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule
Ano meaning ng semi-conservative? Kasali ba lagi or part ba lagi yung old strand as part of the daughter? Yes, okay.
is made up of one old, conserved strand of DNA and one new one.
Ayan. So, tapos na. For a while lang, ah. Ayan. So, overall, guys, natapos natin ang DNA replication in prokaryotes. Okay pa kayo? Ha? Malapit na matapos? So since na-discuss na natin, wait, chat. So since malapit na matapos yung discussion natin, continue na natin agad. After natin ma-discuss ang prokaryotic DNA replication, of course, meron ding eukaryote. Pero sa eukaryotic DNA replication, madali lang din siya maintindihan, may konti lang mga additional. Okay, may konti lang additional. So in DNA replication, take note that DNA synthesis and replication usually happens during S phase, okay, S phase of the cell cycle. And ito yung mga characteristics ng eukaryotic DNA replication. Number one, both DNA parental strands serve as template. Question, sa first characteristic ng eukaryotic, same ba to sa prokaryote? Yes or no?
Yes.
Yes. Okay, very good. Same din siya kasi parehas na strands nagiging template. Parehas ba silang semi-conservative? Yes. Next, it says here, it begins with multiple oris. Same or different? Different. Kasi kapag prokaryote, ilan? One only. That's right. And then it occurs, as I've said, at S phase of the cell cycle. And ang termination process ng eukaryotic DNA replication is not that clear as compared to the prokaryotes. In eukaryotic DNA replication, polymerase also act here. Pero meron tayong mas specified designations ng pangalan nila. Okay? Pero hindi naman nalalayo sa activity doon sa prokaryotes kanina. So for example, dito sa eukaryotes, hindi na 1 at 3 ang term natin. Mga alpha na, Greek alphabets na. Okay? So una, ang RNA primase kanina, dito, polymerase A ang tawag. Okay, polymerase alpha, polymerase alpha. Next, polymerase delta naman ang tawag for the elongation of the leading strand. Okay, polymerase delta. Pero dun sa prokaryotes, equivalent lang ito ng polymerase 3. Okay, DNA polymerase 3. Do you get it? Nagigets? Okay, next. We also have polymerase epsilon. Ang epsilon naman ay designated in elongating the lagging strand. Okay, lagging strand. So both polymerase delta and epsilon, they both possess 3' to 5' exonuclease activity. What's exonuclease? Is it elongation or proofreading? Okay, very good. It's proofreading. And then, polymerase beta naman, this is analogous to the E. coli or DNA polymerase of the E. coli. And this is responsible for excision of primers. So dito, anong equivalent? That is polymerase 1. Okay, tama. That is DNA polymerase 1. So ganun lang. Dadagdagan nyo lang yung terms na na-encounter nyo. Pero yung actions, they are almost equivalent. Okay? So processes, na-discuss na natin ito elaborately kanina, reinforcement na lang. Sa eukaryotes, may helicase pa rin, may single-stranded binding proteins pa rin, may primers pa din, and then take note, it says here, synthesis of the leading strand is carried out by the DNA polymerase delta, while the synthesis of the lagging strand is catalyzed by DNA polymerase alpha. So take note, it says here synthesis, okay? Not necessarily elongation. Next, ito actually pandagdag na lang na knowledge sa inyo. Summary lang din ito ng difference nila. So una, for both prokaryotes and eukaryotes, semi-conservative, parehas sila. May primer na kailangan, and both parental strands can be used as templates. And then, differences, single ang ori pag prokaryotes, Do you still understand? Okay pa? Ha? O, di ba madali na lang siya intindihin kasi na-discuss na natin. Multiple ORIs kapag eukaryotes. And then, DNA polymerase 3 both creates the leading and the lagging sa prokaryotes. Dito sa eukaryotes, may mga designated or specific terms tayo. And then, prokaryotes naman associated with supercoiling. And then sa eukaryotes, both supercoiling and telomere shortening. Okay? So overall, yan po ang DNA replication process. So natapos na natin guys, finally, ang DNA replication. Nagets? Naintindihan po ang discussion? Okay, good. So yung mga succeeding slides dito actually, these are just diseases or factors that could lead to diseases or damage of the DNA. Of course, kapag may damage ng DNA, that could lead to mutation and eventually long-term genetic disorders sa mga tao. So dito, pag binasa nyo, medyo madali naman siyang intindihin. So meron tayong mga causes ng DNA damage like UV light, chemicals, and other agents. And then, ang possible mechanism ng DNA damage could be base alteration. So pag sinabi nating base alteration, the normal complementary base pairs are being altered. As simple as that. So pag mali nga ang pairing, that could lead to disorders. And then, pwedeng removal or loss of nucleotide base. Kulang o kaya nawala. Okay? That could lead to diseases. So meron tayong iba't ibang mechanisms ng DNA repair. So gumagawa ng ways ang paraan to keep process of DNA replication normal pa rin despite of the possible adverse stimuli. So we have mismatch repair, base excision repair, nucleotide excision repair, and double-strand break repair. So pakibasa na lang yung problem and the solution being held by these processes or mechanisms by the body. Okay? Pakibasa na lang siya. And then, in case na hindi na talaga ma-manage or hindi mako-control ang proper repair of damaged DNA, that could of course lead to different diseases. Pakibasa na lang class yung mga iba't ibang klase ng diseases related to DNA repair disorders like Xeroderma pigmentosum. So pakibasa na lang po. And then Fanconi's anemia, yung characteristics ng Fanconi's anemia, and Ataxia telangiectasia. Okay? So yan yung mga iba't ibang diseases related to disordered DNA repair capabilities.